So this is the cartoon of how the alternative complement pathway works. This pathway is completely antibody independent. It is able to be triggered entirely by the innate immune response and takes place both in the innate and the adaptive, but doesn't require anything from the adaptive immune response. It can completely be triggered by the innate immune response. And this is because it is activated by pathogen surfaces, and almost all pathogens activate it spontaneously. These would be things like LPS on gram-negative pathogens, bacteria, peptidoglycan on both gram-negative and gram-positive bacteria, viruses, virally infected cells, even some parasites. All of these have surfaces that are capable of spontaneously activating the alternative pathway. So remember also that the alternative pathway has two extra factors that are unique to it, okay? And these are factor B and factor D, which we're going to see how they function. And note that they are not termed the same way other components of complement are termed that we've talked about in the other pathways. Okay, so how does this work? Well, the alternative pathway begins when C3, the same C3 from our classical pathway, is making its way along and is spontaneously hydrolyzed by water. This literally means that water seeps into the C3 molecule, and in doing so, it hydrolyzes the C3 thioester bond, which it uses to covalently bind to antigen surfaces. What happens is that this causes the formation of a new molecule called C3H2O. This happens at a very, very slow rate. It's happening right now in your own body. That C3 is being converted to C3H2O as water seeps into the C3 molecule. So this C3H2O is able to bind factor B. Factor B is cleaved by another unique component of the alternative complement pathway, factor D. Factor D is always on. It's always active. And when it's cleaved, you get formation of factor BB. In this case, factor BB, the little b here indicating that it is an active component. So once again, we're using this lowercase letter to denote the active form of factor B. Well, this C3H2OBB is one of two alternative C3 convertases. So basically, C3 comes along again, this can cleave it, and then once again, you're going to have C3A and B, just like you would have in the classical pathway. So here's your C3A and B. A is going to head off into the sunset and go away. And then guess what? This C3b, which we're used to from our classical complement pathway, can come in, bind B, B gets cleaved again by D. Now you have C3b, big B, little b. This is another alternative C3 convertase. So this actually sets up another one of those big amplification steps, where then you're just going to have C3b, BB forming in a lot of different places and binding, and you're going to create a lot more of this alternative C3 convertase. Now, as you create more and more C3b, you're doing two things. One, you're creating opportunities for opsonization, right? Because C3b is a really important opsonin. Two, you're amplifying the signal because you've used all these alternative C3 convertases to create more and more C3b, and that is good for increasing inflammation. If one of these C3b molecules binds near a C3H2O, big B, little b, or a C3b, big B, little b, and adding this extra C3b here, I know this is a little complex, but either of these forms what's known as an alternative C5 convertase. So that would be a C3H2O with a big B, little b, with a C3b, or a C3b with a big B, little b, C3b. If you get either of these combinations of 3, you have a C5 convertase. So C5 comes in and then is cleaved, and that leads to the formation of C5a and C5b. At that point, we just go into the final common pathway, which is really the formation of the membrane attack complex, which leads to C5b joining with C6, C7, C8, and C9, causing lysis of the cell. Now, if C3 is constantly hydrolyzed into C3H2O, which constantly leads to the creation of more C3b, how do we stop the alternative complement pathway when we need to? We don't stop it if we want to move forward, creating opsonins and inflammation and bacterial death. But let's say that C3b over here binds to one of our host cells. Well, it turns out that there are 2 types of surfaces in this world. There are activating surfaces, like pathogens, and there are inhibitory surfaces, like our own cells. So it turns out that if C3b lands on an activating surface, or C3H2O for that matter, you'll get the alternative complement pathway as I just laid out. Now, if instead C3b lands on one of your own cells, it will be stopped, and this happens in a couple of ways. Remember earlier I mentioned factor H, or beta-1 inhibitor? Well, factor H actually works to dissociate BB over here from C3b. This basically makes it so that the alternative pathway doesn't happen anymore, because BB then goes away, and as it leaves, it winds up taking away the continued activating stimulus that would create more C3b. The other thing that factor H does is act as a cofactor for binding of factor I. Factor I actually leads to the degradation of C3b into inactive C3b. This one can't actually carry out complement. So you're actually hitting this C3b and BB complex of the C5 convertase in 2 ways. One with factor H, which dissociates BB from C3b, and one with factor I that actually leads to the degradation of C3b. Together, these control the alternative complement pathway. Therefore, it doesn't matter that your body is constantly slowly hydrolyzing C3 into C3H2O, which could set off this whole domino effect, because when it's on your own cells, like this cell over here, these inhibitory factors will inhibit the pathway from moving forward. However, if C3b was to land over here on a pathogenic surface, that would be considered an activating surface, which would allow the alternative complement pathway to take place.